I'm a physicist working in the field of biophysics. I'm interested in how cells get their shape and write programs for numerical simulations of lipid vesicles embedded in three-dimensional space with active forces and curved proteins. Our simulations show that cell shape is governed by few simple but coupled mechanisms. Eukaryote cells have flexible membranes that allow them to have a variety of dynamical shapes. The shapes of the cells serve important biological functions, both for cells within an intact tissue and during embryogenesis. How cells control their shapes and the structures that they form on their surface has been a subject of intensive biological research, exposing the building blocks that cells use to deform their membranes. These processes of shape determination have drawn the interest of theoretical physicists aiming to develop models based on physics. Such models explore quantitatively different possible mechanisms that the cells can employ to initiate the spontaneous formation of shapes and patterns on their membranes. We are particularly interested in one such class of mechanisms, the coupling between curved membrane proteins and the cytoskeletal forces that they recruit. Theory indicates that this coupling gives rise to a rich variety of membrane shapes and dynamics, while experiments indicate that this mechanism appears to drive many cellular shape changes. In the featured research paper, we explored the unrestricted shapes of vesicles that contain active curved membrane proteins in three dimensions using Monte Carlo numerical simulations. The activity of the proteins is in the form of protrusive forces that push the membrane outwards, as may arise from the cytoskeleton of the cell due to actin or microtubule polymerization occurring near the membrane. For proteins that have an isotropic convex shape, an additional protrusive force enhances their tendency to aggregate and form membrane protrusions or buds. In addition, we find another transition from deformed spheres with necklace-type aggregates to flat pancake-shaped vesicles, where the curved proteins line the outer rim. This second transition is driven by the active forces, coupled to the spontaneous curvature, and the resulting configurations may shed light on the formation of sheet-like protrusions and lamellipodia of adhered and motile cells. We found that the presence of the protrusive forces gives rise to the formation of protein aggregates and budding at a higher temperature and lower average protein density, compared to the passive system that is in thermal equilibrium with no active forces. Beyond the budding transition, we found a new and unexpected global shape transition that occurs only in the presence of the active forces. In this transition, the spherical vesicle is transformed into a flat, pancake shape with all the curved proteins and associated cytoskeleton forces around the circular rim. This structure resembles the lamellipodia and ruffles of spreading and motile cells, where the actin polymerization is localized to the highly curved leading edge. Our results show that lamellipodia-like structures spontaneously form when convex proteins recruit the protrusive force of the cytoskeleton on a closed membrane. This mechanism is in agreement with recent experiments providing evidence for the role of such curved protein complexes involving I-bar proteins at the leading edge of lamellipodia. To conclude, our study highlights the rich variety of membrane shapes that may be induced by curved membrane proteins that recruit the active forces of the cytoskeleton. These include steady-state shapes that are not possible for the passive equilibrium system with no active force. The next step is to update our Monte Carlo simulations to include not only isotropic, but also anisotropic inclusions on the membrane. Anisotropy means that these membrane inclusions have a different spontaneous curvatures in both directions. An example of such a protein is the F-bar protein resembling a banana shape. At the Laboratory of Physics at the University of Ljubljana, we are working toward new results with anisotropic inclusions and limitations such as reduced volume of vesicle shapes.